Okay, can we go back to basics? Okay, tell us what a TKI is. Okay.
Sorry, I'm so sorry.
And
yes.
what the role of a TKI is in actually the process of altering those cell divisions.
Yes. So the TKIs are imatinib. Sorry, that wasn't one of the slides, but I didn't actually say that that's imatinib. Or I found out the commercial names like Gleevec or something, which is a bit weird. But anyway, yes. So imatinib is a tyrosine kinase inhibitor, and so tyrosine kinases or all kinases are involved in tightly regulating the cell cycle, and so when you're given your TKIs, it basically prevents the cell from proliferating at a higher rate, and therefore a higher proliferation, you're going to have more of these chromosome errors just because of the rate that it's forcing itself to go through. And so that's a little bit of, the role of imatinib, at least in this context. What does it do? It's a tyrosine kinase inhibitor, so kinases they control the cell cycle. All kinases control the cell cycle. They control your signaling and how they interact with each individual cell. So and a lot of GIST patients or GIST tumours have got this KIT mutation, and that enables cells to rapidly proliferate. And so the TKIs basically dampen down that active on signal, so that it's not as fast. Thanks for your question.
Any more questions?
So I guess the as you've said, the role of the TKIs is to try and dampen down that effect. Do we think that there are any of these chromosome instabilities that are more dangerous, for want of a better word, than others, or do we see a pattern in a GIST, let's say an exon 11 mutation GIST? Do you see any of the chromosomes that have more of an impact than others?
Yes, we would do. I'll speak in the context of just cancer in general, but there's a reason there's a pattern to the chromosome alterations that you have, and that's because not all cancer cells will survive, and the ones that do, it's probably because they've got an alteration or a mutation that means that they're more able to survive in an environment where they are going really fast. They're all, proliferating very quickly. They're using a lot of the resources, the metabolism and all that nonsense. And so, repeat your question, sorry. I've forgotten it. What was I saying?
Yeah, I think the question was: do we see any of those chromosomes, from 1 to 22, that have more of an impact?
And so that we do a lot of sequencing analysis, and some of the stuff that Remus was talking about this morning, that there's next gen sequencing surveillance that we have of patients. A lot of patients have very similar alterations, and actually what we're looking for isn't the pattern that every patient has, it's more the pattern that they don't have, if you know what I mean, to try and pick out why some cells respond to therapies and some of them don't. So yes, there are patterns that mean a tumor is more aggressive. So you have, different oncogenes or tumor suppressors that are particularly bad to have for a or good to have for a cancer cell, for example. But we're more interested in the ones that we don't see as often, I think.
So ultimately, what we'd like you and your colleagues across the world to do is find a way for all cancers of turning off what makes the cancer cell divide and proliferate to the extent that it takes over organs, resulting ultimately in death. And this is one of those stepping stones amongst lots of other stepping stones.
Yes, absolutely.
To try and do that.
Yeah.
Because at the moment, the best that we've got is treatments that, when they work well, turn off that switch to stop it proliferating, getting bigger, to help ultimately big tumours reduce in size because the only cure for cancer at the moment is removal of the tumour.
Yeah.
So the more research and work that you and colleagues, not just in this area, in all sorts of other areas, do, the more chance there is of finding that holy grail, but
Exactly, yeah.
accepting that that holy grail probably isn't just one. There's probably,
yes.
because all cancers have a slightly different way of
overcoming the different treatments and whatnot.
Yeah.
And that's
Yeah.
exactly what I've tried at least to describe to you today, is that this concept itself makes the cancer cells very adaptable and flexible. And so what we want to try to do is make them less adaptable and less flexible, and therefore the TKIs that you have now, the imatinib, they would work not just until you relapse or become resistant, but beyond that, to stop the cells becoming resistant, if you know what I mean.
So I've got another question, if nobody else has for a moment. What does artificial intelligence, what role does artificial intelligence play or could play in this? Because the body is a damned clever instrument, isn't it?
Yeah.
And everything about it is clever. So do you see, or is there any evidence at the moment that AI might come into helping?
Yes, AI is becoming a massive part of research, and I think it's something that we all have to get on board on, no matter how resistant or whatnot we are. Pardon the use of resistance again. Yes, absolutely, because I mean, all of the concepts I've explained here are just one level of it, right? And there's so many moving parts to every part of it. I'm not even talking about the metabolism of the cell or extrinsic things, which is what you were telling me about the tumor microenvironment stuff. And we're going to use AI for those types of things to not only incorporate just these intrinsic chromosome things, but incorporate everything all at once, and I think it'd be a super valuable tool. And like you said, it's a big booming data world, and I think it's an exciting place to be in for research. And I think AI is going to be a big part.
Yeah, because I mean, as I said earlier, we're lucky in that we have a therapy for GIST that can target our cancer, whereas there are an awful lot more other cancers that are dealt with through bombarding the whole body with something that attacks all rapidly growing cells, such as your hair, which is why you lose your hair when you have normal chemotherapy, for example, whereas ours is a targeted growth. So I see, maybe I'm a bit too optimistic here, but I see GIST and the therapies and the treatments for GIST as the second generation of cancer treatment that targets to not only attack that element of the cell that keeps reproducing, but also reduces the side effects for patients that have to go through it. So if we could find a targeted therapy for every cancer, that would be a massive step forward to help, particularly in tablet form as well, like we've got.
Absolutely.
Because at the moment, full chemotherapy, you've got to go into hospital and have it intravenously put into you. So, but yeah, I mean, it's very interesting. It'd be nice to get an update maybe from you as your research
we'll keep in contact.
continues, so that we can tell these guys and others listening, what it is and how effective what you're doing is for us. Because I don't know if anybody knows, but imatinib, which was our first line treatment, does anybody know the history of it? Or put your hands up if you don't know what the history of imatinib is. It's really brief if you don't know. Right, so imatinib was found accidentally. So it was a drug that was being used to treat CML? Yeah, chronic myeloid leukemia. So there was a lady, I can't remember what the year was, 1997, something like that?
I definitely
There was a
don't know
lady who had CML, chronic myeloid leukemia. She was being treated with imatinib, and they did some scans on her, and they found that she had a tumour. And it started to reduce in size. So the imatinib was then tested on this tumour, and they used to categorise GISTs incorrectly without realising it was a separate type of a cancer. So because this TKI started to affect the size of that tumour, they then realised that actually this is a different cancer to the one we're treating, but this treatment is helping that cancer. So that's how imatinib was found to treat that cancer. Yeah, I think it was in Japan. Yeah, so it was accidental. Now, more recently, what that has done, and I've been involved with this as well through the other charity, is that, so are there other cancer treatments out there that could treat other cancers? So there's been a big push and project, and I'm not sure if you've had anything to do with this, on what's called repurposing. So we say, okay, we're treating this cancer with this drug. Does this cancer have any similarities to this cancer that we could try that drug on to see if it treats that drug, to see if it treats that cancer in the same way? And that's called repurposing, because they've already been through all of the regulatory stuff to allow it to be consumed into the human body. But because different cancers, they might be different, but they have some similarities, we might be able to have drugs already out there that can deal with other types of cancer. So rather than just reactively thinking, oh, there's another tumour in that person's body that's been positively affected by this drug, they're actually looking for any potentially repurposed drugs. So that's another positive for the cancer community as well. Any more questions? Good.
Thank you very much. You just talked about chromosome division and the TKIs in inhibiting that chromosome division for deficient chromosomes. From someone who's got an SDHA deficiency, that inhibition won't work because it's a gene that's failed on that. Are there any potential treatments or cures for that apart from stem cells or something like that, which may change the current mutation that that person has in an SDHA that would change their prognosis in life?
Yeah, actually that's something I hadn't even thought of, because at the moment we're just dealing with GIST as a whole, but as I've learnt today, there's so many different variants of GIST, and actually what we should do, and I'll think about this as I go back to my work and whatnot, is to do the types of experiments that we have and do them in each subtype of GIST to understand, because like you say, you might not have chromosome instability. There are cancers that haven't got chromosome instability and their genomes are quite stable. They have mutations and whatnot, but the chromosomes are quite stable in copy number. And so I can't answer your question right now, but it's definitely something that I'm going to think about, is to look at the different subtypes and understand what's going on with them, yeah.
Yeah, sorry, just to follow up on that question, because it is a good question. Yeah, the SDH deficient, there is research that we're funding or jointly funding that's being done at the University of Southampton, which is trying to understand the way that cells, SDH deficient GIST cells, grow or multiply, and why is it that they're not working in the same way. So the TKIs, as you rightly say, are not going to slow something down because it isn't there to begin with. So there is a whole lot of research going on into that area as well.
Just looking at, excuse me, just looking at the graphs that you, the graphics that you had up there. Is that better? Okay. So in terms of the body's immune system response to that instability, is there anything coming out of the work that you're doing that helps us to understand how the immune system could actually be proactive in targeting, recognizing, targeting, and actually dealing with these cancer cells which do have this instability?
Yeah, that's actually a really great question. So there is research, and we do some of this research ourselves, where because of the chromosome missegregation that I'm showing here, the chromosome material is actually not just in the nucleus, but in the rest of the cell. And that signals via this other signaling pathway, which actually is in our bodies to defend against viruses, because your viruses insert their own DNA. And so that actually does activate the immune system. So chromosomally unstable cells, they will activate the immune system, but like I've said, is that there's adaptation there, and they can switch off these pathways that mean that they activate the immune system. So that's another angle that we're looking at as well, to try and prevent that switch off of the immune system, so that, like you said, our immune system can help clear the cancer cells.
Okay, is that it? Any other questions? No, right. Thank you very much.
Thank you very much. Thanks for listening,
Molly.
guys.